Coding for a strand of DNA. Given a strand of DNA, we know that we can transcribe it to become mRNA, and we can do this by replacing all the T's with U's and reading the sequence from the 5' to the 3' end. In our translated amino acid sequence as shown here, AUG is close to the 5' end. We can find our codon AUG by having A in our first position, U in our second position, G in our third position, giving us methionine AUG, also known as our start codon for any mRNA sequence. We can use the codon dictionary to translate all the codons in the sequence until we find a stop codon. Anything after the stop codon does not get translated in the sequence. Now that we have our DNA sequence translated and transcribed, we can begin to see what a mutation can do to our strand of DNA. In our first mutation, we have G getting changed to a C in our mutant sequence, also known as a transversion point mutation. Purine to pyrimidine, G to C in our case. The transversion effect has on the normal sequence is TGG getting transcribed as UGG, translated into the protein tryptophan. In the mutant sequence, we have TCG getting transcribed as UCG, translated into serine, tryptophan changed to serine, and the effect of the mutation is missense because the amino acid has changed. In our second mutation, we have G getting changed to an A in our mutant sequence. In this point mutation, we have purine to purine, or G to A in our case, also known as a transition point mutation. In this transition point mutation, in the normal DNA sequence, we have TGG getting transcribed as UGG, translated into the protein tryptophan, and in the mutant sequence, we have TGA getting transcribed as UGA, and translated into a stop codon. A tryptophan changed to a stop codon. The effect of the mutation is nonsense because the codon that was an amino acid changed to a stop codon, ending the translation early. In our third mutation, in our sixth codon, T was changed to a C in the mutant sequence, also known as a transition point mutation, pyrimidine to pyrimidine, or in our case, it was T to C. The effect of the translation point mutation was TGA getting transcribed as UGA and translated into a stop codon. In the mutant sequence, we have CGA getting transcribed as CGA, translated into arginine. The stop codon changed to an arginine. The effective mutation was read-through because the stop codon was changed to a codon that codes for another amino acids. In our last mutation, we have in our fourth codon T getting changed to a G, also known as a transversion point mutation, pyrimidine to purine, or in our case T to G. The effect of the transition point mutation was GCT getting transcribed as GCU, translated into alanine. In the mutant sequence, we have GCG getting transcribed as GCG, translated into alanine. So alanine to alanine. The effective mutation is silent because the amino acid coded for the same amino acid. Thank you for watching.